enzymes may be free in the cytosol. So like the enzymes involved in glycolysis are soluble enzymes not bound to any cellular membranes. For example, hexokinase. So hexokinase need to remain in the cytosol unbound to any membrane in order to catalyze its reaction. Whereas there are some enzymes which remain bound to the membrane in order to catalyze their chemical processes. For example, adenyl cyclase, and we know this is a signaling protein. This adenyl cyclase is bound to the plasma membrane of most of the living cells, and they form cyclic AMP from ATP. Cyclic AMP is a signaling molecule. It is a second messenger which catalyzes many chemical processes. And one more example is tyrosine kinase. So tyrosine kinases are the membrane receptors. So they are the receptor on one hand, they bind to their signals on outside of the membrane, whereas the other end has a phosphorylase activity which phosphorylate each other to facilitate a signaling process. Hence, enzymes can transduce cellular signals by binding, by remaining bound to the membranes. So, enzymes have multiple cellular localization, but mostly they can be divided into two categories, either membrane bound or free cytosolic form. And depending on their uses, they occur at various places, and their membrane bound or free cellular form are required for different chemical processes.